Hi everybody and welcome to the Harry Carey Show. I'm Harry Carey. Today we have a great show lined up for you today. All the way from Georgian College, we have a student named Natasha Curtis here. Welcome, Natasha.
Thanks, Harry. Glad to be here.
Now, Natasha, they recently discovered that your body makes protein. Now that's got to be interesting for you.
Well, they discovered it a long time ago, but oh yeah, we're thrilled. It's actually called protein synthesis. It is simply the making of proteins. Although the term itself is easy to understand, there's multiple steps that a cell must go through. In order to make one protein, the body must seek the aid of messenger RNA, transfer RNA, DNA, amino acids, ribosomes, and multiply enzymes.
Hey, that's something else. Let me ask you something, Natasha. What is DNA?
Good question, Harry. DNA is nucleic acid that contains the genetic instructions used in the development and functioning of all living organisms and even some viruses.
So I was wrong. I thought it was don't know anything.
Yeah, no, it's actually deoxyribonucleic acid. I have a diagram here that I would like to explain how DNA is actually made up. So your DNA has two main strands that form together to make a helix. The yellow part of the strand is called your phosphate group. The green part of the strand is called your deoxyribose group. Together, they are linked together by basic groups, as shown here in the middle. They are drawn together by hydrogen bonding. There's 4 different types of basic groups that hold your DNA together. The first one is called cytosine, and it bonds together with guanine. The third type is called thymine, and thymine bonds together with adenine.
Welcome back to the Harry Carey Show. Before the break, we were talking to Natasha Curtis from Georgian College about DNA. Now she's going to bring us through the protein synthesis.
Okay, so this process doesn't become too overwhelming. Let's go over some of the basics. There's 3 basic steps of protein synthesis that I'm going to be discussing. The first one is transcription, the second one is translation, and the third one is mutation.
Hey, here's an idea. Let's go through some of the basic steps to protein synthesis.
Sure, that's a great idea, Harry. Let's take a closer look at transcription. In the nucleus, enzymes make an RNA copy using a portion of the DNA. The RNA is then transformed into a messenger RNA. The mRNA leaves the nucleus through the pores and gets into the cytoplasm. Here, the mRNA mixes with the ribosomes, which begins the process of protein synthesis. So basically, in this stage, RNA is created so it can go and instruct the making of proteins.
How interesting.
I thought so.
So mRNA is similar to DNA, but is a copy of the message combined in the gene?
That's right. Alright, Harry, the next step I'll be explaining is translation.
Oh, I know what that is.
You do?
Translation. I have a translator come on the show with me when I have such guests as the Jersey Shore cast, for instance. I never know what they're saying, so she helps me out along the way.
Well, that's a great use for the word translation, but I'm actually referring to translation within your body and within protein synthesis. So it's actually referring to the process by which messenger RNA is used as a template to make the corresponding protein.
Interesting use of the word. Go on.
This is where the ribosomes come into play. They are really good at producing protein and are often termed as the protein factory. The ribosome reads the start codon, AUG, and the tRNA brings the correct amino acid. Then the amino acids bond together and begin forming the protein. Once all of the amino acids have been linked, the mRNA signals stop. There's actually no tRNA code for stop. The ribosomes release the mRNA and the amino acids and the protein have been made. So I actually have a short video that will help demonstrate this a little bit more because I know it's a lot of information all at once. So
Great.
inside your nucleus, this is your DNA, and you can see it unwinding and unzipping itself. The red part is the mRNA, and it's reading the codes. And the start codon for reading is AUG. So as it's reading the codes, it leaves the nucleus, goes into the cytoplasm. Now it's now inside of the ribosome, the top part and the bottom part of the ribosome. So as it's reading all of the mRNA codes, it's now creating your protein. So you can see at the bottom, it's starting to spool out your bits of protein. So as it continues to read, the time passes, and wow, look at all the protein you have. So it will continue to make protein until the stop codon comes. So once that comes, it will release from the ribosome, and now it's a 3D structure. The last thing I'll be talking about is mutations.
Mutation. That's an interesting word.
Yeah, it's kind of like a recipe. When you change the ingredients in your recipe, you change the outcome of what you're actually going to be making. So the first one I'll be talking about is called point mutation. This is a change in the nucleotide sequence of a gene. So do you remember in the beginning when I told you cytosine bonds with guanine and thymine bonds with adenine? So it's a change within that sequence. This can result from an error during replication that is not corrected by the usual repair mechanisms. This can result in a code for a different amino acid or even a stop codon. In frameshift mutation, an insertion or a deletion of a nucleotide occurs. This will disrupt the triplets. This will always alter the gene expression.
Well, that's all the time we have for today, folks. Thank you, Natasha, for making an appearance on my show today.
You're very welcome.
Next week, we'll see if we can get Albert Einstein in.
Harry?
Apparently, Albert Einstein died 58 years ago. We'll see if we can get him anyway. That's all the time today, folks. Till next time, Cubs win! Cubs win!
Thank you!